Hello, my name is Eric Park, and I'm going to talk about how fire alters plant microbiome assembly patterns. So, the plant microbiome plays a crucial role in promoting plant health, enhancing nutrient uptake, and protecting against pathogens. Understanding and harnessing this complex microbial community can lead to more sustainable agriculture, increased crop yields, and reduced reliance on chemical inputs. Additionally, plant microbe interactions contribute to ecosystem resilience and biodiversity, making the study of plant microbiomes essential for addressing global food security and environmental sustainability. The researchers in this article proposed that the microbial community would differ across various habitats and that the impact of fire on microbial composition would vary among these habitats, implying a habitat X fire interaction. Specifically, the researchers anticipated that given the indirect influence of fire on plant microbiomes, endospheric microbiomes would be less affected compared to rhizosphere and bulk soil microbiomes. The researchers conjectured that changes in microbiome composition could be partially attributed to plant and soil chemistry influencing plant health through altered functional roles of microbiomes. Employing source tracking modeling, the researchers also hypothesized that with the decreased microbial abundance in post-fire soil, the significance of vertical transmission in fine roots, stems, and leaves would rise with burn severity. Their overarching objective is to characterize the aspen microbiome and its response to fire, shedding light on microbiome dynamics under the influence of disturbance. Given the novelty of this research, their findings contribute fundamental insights into plant microbiome community assembly, offering potential benefits for leveraging microbial communities to enhance plant and ecosystem regeneration. So understanding the post-fire regeneration of vegetation is imperative to mitigate ecological impacts and hasten ecosystem recovery, especially with the increasing size, severity, and frequency of wildfires. The plant microbiome, composed of microorganisms within or closely linked to plant tissues, plays a vital role in ensuring plant health and survival by providing resistance to pests, fostering plant growth, and supplying essential nutrients. Despite acknowledging the significance of the plant microbiome, there is a current knowledge gap regarding its assembly in post-fire environments. This gap is particularly pertinent for quaking aspen, as you can see on the top right, which is a widely distributed species in North America renowned for supporting diverse wildlife and understory plant species. Aspen, dependent on fire for regeneration, primarily reproduces through root-associated suckering after intense fires. Exploring the aspen microbiome post-fire has the potential to shape management strategies that improve the sustainability of this crucial plant species. So for methodology, the research was carried out within the Fish Lake National Forest situated in central Utah, USA. Key tree varieties in our research zone comprise trembling aspen, subalpine fir, Douglas fir, and Engelmann spruce. The soil types belong to Sessions, Fame, Embargo, Elwood, Claiborne, and Bickmore families, characterized as agricryals. The location of this study ranges between 2,900 and 3,000 meters in elevation, with an average annual temperature of 4 degrees Celsius and precipitation of 762 millimeters. The Fish Lake National Forest conducted a planned wildfire across 2,000 acres 12 weeks after that fire. Regenerating aspens were about 0.5 to 1 meters in height. Thus, plants and soils from 9 5-meter radius plots from each burn severity level were sampled over 3 sites. At each plot, the researchers sampled and composited plant tissues for 3 less than 1-year-old aspen saplings. They used DNA extraction, PCR amplification, and sequencing on the samples to find the relationships between microbial community composition and soil and plant chemistry. They sent frozen samples to another lab for analysis. Okay, so for results, alpha diversity. So in this graph, we can see that both habitat and burn severity significantly affected archaeal and bacterial alpha diversity. Among the unburned samples, habitat emerged as a determinant of archaeal and bacterial alpha diversity. Notably, soil and rhizosphere habitats exhibited approximately 2 to 6 times greater diversity compared to compared to plant endosphere microbiomes. After analyzing the differences individually at Q=0, it was found that burn severity only significantly affected leaf archaeal and bacterial alpha diversity, indicating a 40% reduction in diversity within burned plots compared to the unburned counterparts on average. However, at Q=1 and Q=2, they failed to find differences in leaf archaeal and bacterial alpha diversity among burn severity levels. For fungi, we can see that the habitat and burn severity interact. Among the unburned samples, we can see that habitat is also a determinant of fungal alpha diversity, as soil and rhizosphere habitats were 3 to 5 times greater in diversity than the plant endosphere microbiomes. Due to the interaction between habitat and burn severity at Q=0, burn severity only significantly affected fungal alpha diversity in below ground habitats. There are major differences between high severity and unburned plots. Let's see. So at Q=1 and Q=2, burn severity only significantly affected leaf fungal alpha diversity. For microbial community composition, habitat accounted for 26.1% of the variation in archaea and bacteria community composition. Due to an interaction between habitat and burn severity level, researchers analyzed each habitat separately. Within leaf, rhizome, rhizosphere, and bulk soil microbiomes, differences were evident at the phylum and class level, as you can see, with higher relative abundances of firmicutes and gamma proteobacteria in fire-affected samples. However, in the rhizome, taxonomic differences were observed at the genus level, with higher relative abundances of Pseudomonas in burn samples and Streptomyces in high-severity burn samples. Archaea compromised a small portion of reeds in our dataset and decreased in relative abundance in rhizosphere and bulk soil microbiomes with increasing burn severity. Spatial autocorrelation of the archaeal and bacterial microbiome composition was not detected among habitats at each burn site, indicating independence of plots within sites. For fungi, habitat influenced 16.3% of the fungal community composition. Due to habitat and burn severity interaction, the researchers analyzed each habitat separately. They found differences in burn severities only in leaf, rhizosphere, and bulk soil fungal communities. These differences occurred at different levels. At the phylum level, more severe burns led to shifts in certain fungal groups. At the family level, certain fungi associated with plant pathogens increased after fires, especially in leaves. And at the genus level, changes in the dominant fungi in bulk soil were observed with increasing burn severity. Spatial autocorrelation was only found for the fungal rhizosphere community in the unburned site. So microbiome composition and plant chemistry. The soil pH was higher in the high severity burn treatment compared to the unburned site in both the rhizosphere and bulk soil. Fire didn't notably affect carbon and nitrogen levels in the bulk soil or in the rhizosphere. However, inorganic nitrogen concentrations increased with higher burn severity in bulk soil. Soil chemistry differences were associated with the composition of archaeal, bacterial, and fungal communities in both bulk soil and rhizosphere, explaining a significant portion of their composition. In the bulk soil, pH, total nitrogen, and ammonium were significant factors for archaeal and bacterial communities, while for fungal communities, all factors except pH were significant. Similarly, in the rhizosphere, soil pH, carbon, and nitrogen were significant factors for both archaeal, bacterial, and fungal communities. Elemental concentrations vary across burn severities among different plant tissues, with notable differences in leaf tissues, where nitrogen, phosphorus, and manganese concentrations increased, while potassium and calcium concentrations decreased with burn severity. However, unlike soil microbiomes, the composition of plant tissue microbiomes generally didn't relate to the chemistry of the plant or organ, except for the fungal leaf microbiome, where half of the variation was explained by the leaf tissue's elemental composition, although none of the elemental concentrations were significant predictors individually. Of potential pathogens, the researchers categorized fungi into groups, finding 55 arbuscular mycorrhizal, 554 ectomycorrhizal, and 243 potential pathogen ASVs. Potential fungal pathogens were most abundant in above ground plant parts, but how fire affected them depended on the habitat. They looked at each habitat separately and found that only the leaf microbiome has more potential fungal pathogens in high severity burn areas, especially genera like Cladosporium and Alternaria. Fire changed the types of fungi in the ectomycorrhizal communities, but didn't affect the overall numbers of diversity across habitats. Interestingly, the impact of fire on specific fungi varied across habitats. For example, Wilcoxia and tuber increased in fine roots with fire, but decreased in bulk soil, while Rusula decreased in fine roots but increased in bulk soil. Fire reduced the abundance of arbuscular mycorrhizal fungi in bulk soil, but not in the rhizosphere or fine roots. However, there weren't many arbuscular mycorrhizal fungal types, so researchers couldn't thoroughly analyze their diversity. A microbiome source tracking. In the microbiomes we studied, we could identify the source for about 20% of the archaeal, bacterial, and fungal communities on average. The influence of a burn severity sometimes changed how much each source contributed to these communities. Specifically for the archaeal and bacterial communities and stems, and for the fungal communities and leaves and stems, the contribution from rhizome-derived archaea and bacteria increased. Also, while the rhizosphere was a significant source of fungal taxa for leaves and stems in unburned areas, its contribution to these microbiomes decreased significantly in burned areas. However, the source contributions to the archaeal and bacterial communities in leaves and fine roots, as well as the fungal community in fine roots, remained consistent across different burn severities. Okay, so contrary to the researchers' hypothesis, it was difficult to relate the specific chemical changes within plant tissues to the direct impacts caused by fire on plant microbiome composition. This study reveals that fire affects not only the soil and rhizosphere microbiomes, but also extends its impact to the broader plant microbiome. While previous research has shown the soil microbiome's response to fire, these findings go further by demonstrating changes in certain plant tissue microbiomes due to fire, influencing both the diversity and composition of these microbial communities, microbial communities. Although some plant habitats showed no response to the fire, we observed that fire had a significant impact on the composition of archaeal, bacterial, and fungal communities in the leaf fillet. Surprisingly, the effect of fire on the leaf microbiome was even more pronounced than on the rhizosphere or bulk soil microbiomes. This finding was unexpected because leaves emerged after the fire, suggesting that their microbiomes did not directly experience the fire's disturbance. These results underscore the importance of considering the indirect effects of fire on the plant microbiome, similar to what has been observed in soil ecosystems. So for conclusion, prior research has not gone into the comprehensive impact of fire on the whole plant microbiome. Therefore, these discoveries offer essential insight into how plant microbiome communities are formed and influenced by disturbances. Overall, these findings highlight a significant influence of fire on the aspen microbiome, with variations observed between microbial populations in both the plant and soil habitats. Future research into how the plant microbiome reacts to fire should focus on extensively measuring the plant metabolome response, along with examining variations in site features like solar irradiation and temperature between areas affected by fire and those untouched by it. All right, so these are the questions. So the first one being, should researchers conduct planned wildfires for these type of studies? If not, what other method would you recommend? Do you think it was right for them to start wildfires for the experiment? The second question is, how does the frequency and intensity of wildfires impact the composition and functioning of soil microbiomes, and what further research is needed to fully understand these effects? The third question is, how do archaeal and bacterial communities compare to fungal communities and plant microbiomes, and what are the differential impacts of fires on these microbial groups? The fourth question is, which figure do you believe is most important and why? Which figure, if any, needs improvement and how? Last but not least, what can the researchers do to improve their paper? Are there any more new methods that they could use, or can they try using different new species? And here are the references. Thank you.